All right. How are we with time? Well, I think we can start now. Good afternoon, everyone. I hope you had a nice lunch and that we have now energy to continue with our conference. So now we will start with the session, which is a bit special. That is why we call it poster specials. And I would also like to ask for all the people who are poster presented in the regular poster session too, to be prepared, because immediately after the poster specials, we will go to the poster presenters and discussion. We yesterday had a very nice poster presentation with a long, interesting discussion. So we would like to today continue the same way with the poster session too. Before we begin, I just want to say that the voting for posters is already open. You can, you could have voted since yesterday, but I would really like to give you please view all of the posters before voting. The voting is going to be opened until 5 p.m. Today, Japanese time. So at that time, at exactly 5 p.m., we're going to close the voting and see the number of votes.
And the voting for the assembly.
For this, yes, also for the, you can also vote, still vote for the assembly topics. Okay, so in this poster specials session, we are going to have 3 posters, which the presenters are going to also present orally. And we will start with the Siti, Anis, Dalila, Muhammad, Zahir. Anis, you're here, I see. So Anis is from Malaysia University, and she belongs to a group of Professor Omar, who is doing a great work in aquaphotomics for a number of years now on very, very different topics, including medical diagnostics, including food preservation and so on. Anis will today present her poster, and she also wrote a paper on this topic, which will be published in our journal, first journal issue. And today if you can please, Anis, present your topic in like 12 minutes, and we will have some questions afterwards.
Right. Thank you, Dr. Jelena. So my name is Anis, I'm from Malaysia. So first of all, I would like to express my gratitude for giving me chance to be here. I'm actually very, very new in this study. So these past 2 days had been really inspiring for me. And so can I share my screen, because I prepared like—
Yes, please.
Yeah. So this is the poster that I prepared, and I actually provided the figures in slides so that it's easier for us to see. So we'll go through this one by one. So my title is about near-infrared spectroscopy and aquaphotomics in Carambola beta and Averrhoa. So a brief introduction about Carambola. It is also known as star fruits, and the main composition consists of up to 91% of water. So because the main composition is water, so that's why I think it is quite interesting to do the aquaphotomics data analysis on these samples. So as you can see here, the Carambola fruits is classified into 7 grading indexes, and it is according to the ripening stage of the fruits. So this grading index is actually provided by the Malaysian Federal Agricultural Marketing Authority. So you can see here from index 1, the carambola is still immature and dark green in color, and the best time to consume these fruits is at index 5. So as the peel of the fruits changing color, the pigments on the fruits such as carotenoids, chlorophyll, and also anthocyanins actually affect the distribution of light on the fruits. So in this study, we're going to see the water absorbance bands corresponding to the Carambola index, pH value, and also the soluble solid contents. So apart from that, we're also going to see 2 different spectra measurement as a comparative analysis, which is the interactance method and also the reflectance method. So this is the methods that we use in this research. So in this study, we use a total of 50 samples at different maturity levels. And the first thing that we did was to measure the weight of the samples, and also the the firmness value by using the penetrometer. So once all the data is recorded, we did a NIR spectra measurement using 2 measurement methods, which is the interactance method and also the reflectance method. So the main differences between these 2 methods are the configuration of the fiber probe and the calibration system. So basically, the reflectance technique was conducted with a probe of 6 illumination fibers surrounding 1 reed fiber. Meanwhile, the interactance technique used a combination of a reflectance probe and a single strand fiber. So the calibration technique for reflectance and interactance were different in terms of the distance of the probe to the white from the white reference. So, but during validation measurement, both techniques require the probe to be perpendicular to the surface of the samples. So after non-destructive measurements using spectroscopy had been conducted, we extract the juice from the samples and use it for pH and also soluble solid contents measurement. So moving on to the data analysis, I tried to calculate the mean value of soluble solid contents, pH, and also firmness for each samples according to index, and then we could see here this is the absorbance spectra for both methods, and we can say that as carambola ripen, the fruit become less firm and high in moisture content, and also the value of pH and soluble solid contents increases as well. So these figures shows the absorbance spectra of the fruits after smoothing spline. So in this study, we used a local quadratic regression using second degree polynomial as an approach to fit curves and surfaces to the data. So you can see actually both method shows a curve at 970 nanometer, but when we combine these 2 methods in one graph, we could see that the interactance method show a more distinctive curve compared to the reflectance method. So the bottom peak that lies at approximately 970 nanometers shows the correlation of NIR spectra with the OH functional groups. So next, I also tried to construct the aquagram according to index, pH, and SSC value. We could observe a variety of patterns that are trying to give information towards the interaction. So basically, the changes in this pattern are due to the changes of free water and hydrogen bonding water molecules, and also the differences in the concentrations of the chemical substances in the system. So from this diagram, we categorize the Carambola into pH of range of 2, 3, and 4, and a high absorbance band could be observed in the Carambola of pH range 2, and this aquagram shows that as the value decreases, the pH level increases and the fruits ripen. So from the aquagram of the interactance method, we could observe that the pattern started to change at the wavelength of 970 to 1025. So the highest absorbance can actually be observed at 1025 nanometer. Next, in this figure, this is the aquagram categorized according to soluble solid contents in the range of 5 to 9. So this pattern shows that as the value of the soluble solid increases, the aquagram value decreases. So as the solid content increases, the water absorption bands are perturbed, and this phenomenon indicate that the spectra of the water are influenced by the interaction with molecules in the samples, and also a high absorbance could be observed at 1100 nanometer in the reflectance measurement. So the maximum absorbance in the range of 1100 could be influenced by the stretching of OH molecules and also the increase of the hydrogen bonds. So the increase in hydrogen bonds is implicated by the increase of the concentration of the solid content like sugar. So since the changes in the spectra of water caused by perturbations are very subtle, it is very important to find a suitable method that is capable to extract useful information related to this study. So by having both measurement methods as a comparative analysis, small changes in the water spectra could be analyzed in detail. And from both methods, the interactance method shows a higher accuracy and precision for spectral analysis. And like this study actually was also conducted by my supervisor, and he supported that he stated that the interactance produced more precise results due to lower differences in the intensity of light during the measurement of calibration and actual samples. So to summarize, this study highlighted the differences of the interactance and reflectance techniques in spectral data measurement of SSC, pH, and also firmness of Carambola of different index. So the value of the pH shows the higher accuracy in both calibration and prediction method. So this is because NIR spectra are highly sensitive to pH, and we can conclude that the wavelengths in the range of 970 and 1100 could provide information on SSC, pH, and also firmness of the fruits. Thank you.
Thank you, Anis.
Yeah.
Okay, so we are now, we will have a short time for a few questions now for Anis. And Anis, please make sure to stay here later to join the post session for other people who would have maybe questions. I would like now to ask the audience, do you have any questions for Anis? Well, in that case, I have a question. First, a comment. I really like that you're doing the trying to correlate the firmness value. This is something we currently don't have the instrument, and I definitely wanted ordered to connect these two things. I wanted to ask you, how did you calculate the aquagrams? Because in my opinion, you have a very tiny difference. So I think that can be enhanced if you use some tools. Can you please explain?
I use the standard normal variation method to calculate the aquagrams.
SNV.
Yeah.
I see. Okay. Well, I think in this case you have a very, very large difference of coming from the physical information, basically, that it should be corrected. It could be corrected. I mean, it says something about your samples, definitely, about the physical properties, but you can definitely go into more depth. Professor Tenkova has a question.
Yes,
I think.
I just have a comment. When I look at the spectra, the y-axis is absorbance. And usually water has absorbance at 960.
960.
So it is transmittance, probably. So in order to calculate absorbance, you have to follow the formula of Lambert-Beer law, logarithm of I0 over IT minus. So and number one, so just be careful in calculations and transmittance and absorbance. And when you write Aquagram, you just rescale, because now Jelena pointed out the small differences. If your maximum of the Aquagram is on the left side -4, then you can better see the differences. This is what I mean. It's just a matter of technique. Otherwise, the work is very interesting, and it is very important to know what are the water species and water bands related to solubles in this particular fruit, and especially what is the process of ripening. So if you can monitor this process to see which are the species that are changing with ripening, and it will be very helpful to when you have a fruit and you take a spectra, you can say which stage is this. I think this is your final goal, I can guess, from your presentation. Thank you very much.
Yeah.
Anis, you have a comment and a question from, yes, Harpreet and Zoltan. First, Zoltan. Zoltan? So, okay, maybe there is a problem with connection. So, Zoltan says, thank you for the nice presentation. What was the wavelength range, the whole range of the instrument that you used? And before the pre-treatment, he says you need to truncate to do the more narrow range. That is his opinion. So,
Right. Should I answer now?
Yes, please. Yes, yes.
Yeah.
Yes, we have.
Thank you for your comments. So it's really helpful for me because I'm very new, right? So I use the wavelength range of 800 to 1100. Yeah.
The whole range. Okay.
Yeah.
And there is a question from Harper's Crowler. She's the. I wanted actually to mention for you to connect because you're working on similar topics. Yeah. She has Harper's actually. Can you ask if you want to share your screen? Ask yourself.
Yeah. Actually, my question is how the water bands were calculated to plot the aquagram, because I can see the resolution of the aquagram. I mean, the difference between two water bands wavelength is 25 nanometers, and it's like it's consistent across the whole water band wavelength. So how the water bands were calculated? So
actually before this, I was not very clear about this, but because I did this before the conference, right? So yesterday after I heard like the talk from Dr. Harpreet, I found that I could improve more in this part. So yeah.
So yeah. Harpreet, thank you for the presentation yesterday then. Good.
Thank you.
Any other questions? Okay,
This
so
is how
Anis,
it
please
goes, yeah.
just stay here. We will continue later for more discussion. So our next two presenters are coming from one of the sponsors of the big sponsors of this conference, and our collaborator and supporter over many years. The presenters are coming from the